A couple of things to just kind of catch up. If you didn't get done with everything, I kind of put the materials over here that you might still need. The slides, the micrometers, tested sterile water, paper towels, swabs. There's some other plates over there if you need other plates. Tomorrow we'll be doing exercise number 2, which isn't nearly as lengthy. Basically, you're going to practice aseptic technique, transferring microorganisms from one kind of media into another. So we'll transfer them from a broth culture into a sterile broth culture, from a broth to a slant, etc. Just getting you used to using your inoculating loops and practicing good sterile technique. And so there's only 2 or 3 pages to read, but it will be helpful to read that ahead of time so you have an idea of how the techniques work, if you haven't done it before. Okay? I understand we're still having issues. I had been told that the website was back up and running again, and someone was saying we still can't get stuff there. So did you all get Natalie's Dropbox invitation? And I was able to send also via just email one PowerPoint and one lecture to Kayla. It took a while to get through, but it did get through. So if you have issues with Dropbox and getting stuff, let me know. But, hopefully we'll get this resolved.
Did you ever figure out if there's any stuff you can do on Mastery Micro? Yeah.
I'll work on that this afternoon. There will be some stuff, yes. And I know that there's something for cell wall structure that we can look at. There'll be some things for optional points, but let me work on that. On the other hand. Okay. Alrighty, so we were talking about cell wall structure, and particularly in the context of gram-positive, gram-negative cells, how their cell walls differ from one another structurally. And we were in the process of talking about the gram stain and how the outcome of the gram stain reflects these fundamental differences in gram-positive and gram-negative cell wall structure. So where we left last time was we had added the primary stain, the crystal violet, which stains both cells, gram-positive and gram-negative, purple. Adding the iodine just kind of precipitates the dye crystals, particularly in the gram-positive bacteria, because the peptidoglycan layer is much thicker. Much less effect of iodine on the gram-negative cells because of their thin peptidoglycan layer. So really, the mordant is kind of designed to keep the primary stain in the cell walls of the gram-positive bacteria. Particularly when you do the next step, and that is the decolorization step, involving 95% ethanol. And this is the critical step in the gram staining procedure. Because what you want to do is you want to rinse those cells with alcohol just enough that it washes out the unbound crystal violet from the gram-negative cells, but it's retained in the cell wall of the gram-positive cells. And particularly that mordant helps to retain it in the gram-positive, thicker peptidoglycan layer. So if you decolorize properly at the end of the decolorization step, the gram-positive cells are still purple and the gram-negative cells are clear. Okay? Now, can you decolorize too long and completely wash this out too? Yes, you can. Can you decolorize for too short of a period of time and not get the purple out of the gram-negatives? Yes, you can. So one of the things that we typically will do, especially initially until you get your technique down, is when we do a gram stain, like say on your unknown, we will on that same slide have 2 additional organisms, one of which is a gram-positive control and which is a gram-negative control. And so if you're treating this whole slide the same, if these come out purple, these come out pink, then you're confident about the outcome of your unknown. Okay, so I would kind of encourage you to get in the habit of that, particularly early on. By the time you're at the lab exam, you're not going to need these controls. You're going to know the Zen of how long you decolorize. Initially, it's set up to just be a certain amount of time, but that really varies depending upon the thickness of the bacterial smear that's on the slide. If it's real thick, it's going to take longer to decolorize. If it's real thin, then it might take less time. So it's the kind of thing you have to develop, just kind of a little artsy technique, but you will get good at it. Now, why don't we just stop here? Why don't we just go purple and clear? And we add the fourth step of adding saccharin, which we call a counterstain. All three successes. Yeah, it's really hard to see these clear cells. Okay. Both of these cells take up the red dye, but the purple kind of masks the red. So there's both purple and red in here, no effect. Over here, when these cells have become decolorized, then they pick up the red stain easily and they have that kind of pinkish color. Okay? Now, for cell wall structure, there are some exceptions to the rule that basically things are either gram-positive, thick peptidoglycan layer cell structures, or gram-negative, thin peptidoglycan layer structures with an LPS layer. One of the exceptions to the rule is that some bacteria, particularly the genus Mycoplasma, doesn't have a cell wall. Doesn't have a peptidoglycan cell wall. This is very unusual. But as you probably learned in biology classes, there are always, typically in living systems, exceptions to the quote-unquote rules. Okay? So this is unusual. What would the consequence be for these cells not having a cell wall? You've got to worry about their environments. Okay, they would be much more prone to be lysed in hypotonic solutions. One of the things that we're going to see in a little bit is that typically the cell membranes in prokaryotic cells lack those sterol molecules like cholesterol. Okay, we have cholesterol in our cell membranes. But prokaryotic cells typically don't. What do sterols do? They help to give membranes more rigidity, make them a little bit less fragile. Okay? So, mycoplasms lacking a cell wall are one of the very few bacteria that do have sterols in their cell membranes. So it's kind of a compensating factor. Okay? In addition to protection from lysis, what else do cell walls do to bacterial cells? Remember that I said that all cells without a cell wall are round, right? But then, in reality, some of them are spirillum, some of them are bacillus. They help to define their shape, right? Mycoplasms tend to be pleomorphic. What that means literally is that they can have a variety of shapes. And so if you look at this micrograph of mycoplasms right here, you can see that not all of them look the same. Okay? There might be little small differences in their microenvironment that results in different amounts of water being taken up by the cells. But since they don't have that cell wall around them to define their shapes can be highly variable. Okay, so that's another thing that tends to characterize these mycoplasms. You're not going to have any mycoplasms for unknown. That won't be a problem. Another exception to this basic cell wall structure is found in the genus Mycobacterium. And we will work a little bit with Mycobacterium. There are two pretty well-known species of Mycobacterium, Mycobacterium lepri and Mycobacterium tuberculosis. What do these have in common? They sound like diseases, don't they? Leprosy and TB, right? Yeah, these are pathogenic microorganisms. These do have a peptidoglycan cell wall, so that's not the unusual part. They've got the cell wall. But these particular bacteria, particularly these two species, the cell wall is impregnated with a substance called mycolic acid. And this is kind of a fatty, waxy substance. Used to be known as cord factor. Not sure what the etymology of that is, but that's an old-timey term for this mycolic acid substances in the cell wall. The reason that I mention this is that there is a staining test that we can do and will do called the acid-fast stain. And when you do this particular acid-fast stain, if the cells have this mycolic acid in them, at the end of the staining procedure, they turn red. If they don't have the mycolic acid in them, then at the end of the staining procedure, they turn blue. So a very distinct response, depending upon the nature of their cell wall structure. So this can give you a pretty fast and dirty test, particularly for TB. Have somebody hack up a sputum sample, make a slide out of it, do a quick acid-fast stain, and these bacteria are bacillus, and if you get the red staining bacillus cells, then that is highly indicative of this particular genus. And if you couple that with other symptoms like somebody's hacking, coughing, fever, etc., then that's kind of a presumptive indication of the possibility of tuberculosis. Okay. So this particular staining test is fairly characteristic of this particular genus. So that's one of the reasons that we'll do this test, to allow you to quickly eliminate a possible genus for your unknown. Other exceptions to the basic cell wall structure are found in the domain archaic. Now, if you remember, the method of classification that we're going to use breaks all living organisms into three domains: archaic, prokarya, and eukarya. Okay? Eukarya do not have a peptidoglycan cell wall. Plants have cellulose cell wall, but no peptidoglycan. Prokarya, with the exception of mycoplasms, have that peptidoglycan cell wall. And archaea are kind of bizarre. They don't have a peptidoglycan cell wall. But they do have a supporting structure around them that serves the function of a cell wall. And we even call it a cell wall. The chemical composition of that is highly variable depending upon the species of archaean bacteria you're talking about. In some species, it's entirely made out of protein. In other species, it's primarily a carbohydrate structure. In some species, it's kind of a hybrid of protein-carbohydrate. But it's not that characteristic peptidoglycan structure that's associated with the prokarya domain. And then finally, worth mentioning is some bacteria are considered to be L forms. L forms oftentimes will all of a sudden lose their cell wall during stages of their life cycle. So sometimes they'll have a cell wall, sometimes they won't. So if you're doing a gram staining, and one time you get a gram-positive result, and then the next time you get a gram-negative result, your first inclination would probably be, oh my God, I've contaminated things. I've really screwed up, or my technique's bad. but you run your controls and everything's working properly, okay? If you don't have a cell wall, you will stain gram-negative, correct? Because if you look at your gram staining procedure, over here, you apply the crystal violet, both stain purple, and the iodine, both purple. Decolorize, that's when you clear things out. Okay, but if you don't have a cell wall, you're going to be decolorized immediately. So you would come out as a pinkish cell if you lack the cell wall. Most of these archaeans that don't have the peptidoglycan cell structure also will stain gram-negative, even though they have a kind of pseudo cell wall. None of you have L forms as an unknown. Okay, that would be really mean. Okay. However, some of you will have unknowns that fall in the genus Bacillus. And Bacillus are notorious, at least some species are, for being gram variable. What that means is when you do the gram stain, most of the bacillus end up staining gram positive. But in that same population on the slide, some will look lighter in color, almost pinkish in color. And again, if you have a gram variable, one of the first things you're going to be asking me is this contaminated? Okay? So just be aware of that possibility, that if you have bacillus, then it's likely that it may be a gram variable situation as opposed to contamination. But you can check with me on that, okay? That's usually not an issue with other genera. It's primarily a characteristic of the bacillus genus.
Is there a reason for that? Is the cell wall just different?
You know, I really haven't seen, Sam, an adequate explanation for what causes gram variability. Somebody wants to do a little digging. Optional points, okay? You can always suggest to me if I say something that piques your interest. Ask me if you can do a little research into something and we'll negotiate some possible optional points, and so I'll keep that in mind. Okay, so we're talking about the cell envelope right now, and we spent all of our time talking about the cell wall. Let's move further inward toward the cytoplasm of the cell wall. And take a look at the cell membrane a little bit. Cell membrane of a prokaryotic cell is very similar to the cell membrane of a eukaryotic cell. It's got its phospholipid bilayer. It has proteins, many of which are transport proteins. A lot of enzymes associated with the cell membrane of a prokaryotic cell, including things like ATPases. Can bacterial cells carry out aerobic cellular respiration? What do you think? Yes or no? They can. Okay. And they can produce ATP by chemiosmosis. What do you need for chemiosmosis to work? Remember from cell biology? Remember our rows of chairs in the lecture room next door represented the inner mitochondrial membrane? And those of you sitting in the chairs in the middle were in the matrix of the mitochondria. And then that space between the chairs and the walls was that inner membrane space. And you build up a hydrogen ion gradient across that membrane. And then if you have that ATPase channel protein, as the protons flow through that channel protein, that's coupled with ATP synthesis. Remember that? Okay. Well, bacterial cells can do the same thing. But do they have mitochondria and mitochondrial membranes? No, they don't have membrane-bound organelles. So where do they set up their hydrogen ion gradient? Across the plasma membrane. So they develop a high concentration on one side of the membrane, a high concentration on the other side, and then they've got the ATPase channel proteins in their plasma membrane. And they can use that to power chemiosmosis to make ATP. That hydrogen ion gradient I mentioned could be used to power flagellar rotation. Okay? So a lot of the things that are typically associated with organelle membranes in eukaryotic cells have to be associated with the plasma membrane in a prokaryotic cell. It's not also uncommon to see the cell membrane enfolded into the cytoplasm of the cell, oftentimes in kind of a spiral arrangement. This particular structure is called a mesosome. What's the significance of the spiraling type of arrangement of that membrane, you think? What should you immediately think when you see something like that?
Surface area.
Increased surface area, okay? And it kind of makes sense because if a lot of the enzymatic activities are associated with the cell membrane, let's give ourselves some more membrane surface for those activities to happen. Okay. Sometimes those little enfoldings will break off into free-floating membranes. And in photosynthetic bacteria, chlorophyll molecules will be associated with those membranes. They're kind of equivalent to the thylakoids in the chloroplast. Okay? But is this considered to be a chloroplast? No. It's not bound by another outer membrane separating those structures from the cytoplasm. Also, this doesn't have its own DNA. This doesn't have its own ribosomes like chloroplasts do. As I mentioned, typically cell membranes of prokaryotes, with the exception of mycoplasms, lack those sterols, primarily cholesterol molecules. It's really not necessary for them to have that rigidity factor because they've got the cell wall, which really helps to protect them instead. The lipids that make up an archaean membrane are a little bit different from those that make up a typical prokaryotic membrane. Both of them have phospholipids. Both of them have a phospholipid bilayer. In prokaryotes, the bonds that hook together those glycerol and fatty acids to make phospholipids are ester bonds. Remember, acids, carboxylic acids. In the archaeans, the phospholipids are joined together by bonds that are ether bonds as opposed to ester bonds. And these are a little stronger or a little bit more stable than the ester linkages. So the phospholipids in the archaean membranes are a little bit more able to withstand stress, particularly extreme heat and salinity. And that is consistent with the fact that many of these archaeans are oftentimes found in very extreme or hostile environments. I think I mentioned the word last time. Sometimes the archaeans are referred to as the extremophils, the ones that might be growing in the Dead Sea or boiling hot springs or things. So again, adaptations for survival at the molecular level, in this particular case associated with their cell membrane organization and structure. A little bit different way to join the phospholipids together than you typically see. Okay, let's dive into the cytoplasm now. And one of the cellular structures that we're going to be very interested in that's found in the cytoplasm of some bacteria, but not all, are endospores. What does endo mean? Within, inside, okay? So these are structures that are found within the cytoplasm of the cell. The cell that produces the endospores is oftentimes referred to as the vegetative state, or the vegetative cell. In Berge's manual, you will see reference to the term sporangium. Some of you have had botany already, right? You learned about sporangia. Those are structures that house spores. So the vegetative cell, if it produces an endospore, actually is a sporangium. And you might see something to the effect in Berge's manual when it's describing a particular bacteria, is it might say the sporangia is swollen. What that means is the cell is swollen, particularly if the endospore is big, because sometimes big endospores will just kind of distend the cell and it won't be regular in shape all the way around. And so when you see reference to sporangium, it's referring to the cell. Okay? Anybody know what the purpose of endospores are? Okay.
Is it to help us survive our condition?
Certainly as a survival mechanism, and it typically is initiated, if it can happen, by extreme changes in the environment. A depletion of nutrients, an increase in temperature, exposure to radiation. Okay? So all those kind of things that would typically kill a vegetative cell. Endospores are highly resistant to those particular extremes. Now, there are limits, okay? But for example, I mentioned earlier on that vegetative cells are killed by boiling. 100 degrees centigrade is sufficient to denature the proteins and cells and kill them, meaning they can't reproduce anymore. But boiling water does not inactivate an endospore. And given the right conditions, this particular endospore can germinate and basically give rise to a new bacterial cell. It takes a temperature of 121 degrees Celsius, which you achieve in an autoclave, to kill the endospores and prevent them from germinating and doing this. Is the endospore a means by which bacterial cells can propagate in numbers? No. One spore produces one new cell. Okay? So it really is a way to kind of wait out the harsh environmental conditions, and then when things get better, if they do, those particular cells can then reform from that resistant structure. So they're highly resistant to radiation, high salinity, high temperature. How long can these things last? Long time. There are a lot of interesting reports. They've dug up tombs in Egypt, for example, and gotten endospores out of 3,000-year-old mummies, which have germinated. They have found, reportedly, viable endospores from a 25 million-year-old fossilized bee. That's some longevity. Or a 250 million-year-old salt crystal. So those are very extreme, but endospores can lie there for years, decades, and still, under the right conditions, become viable. It's really important to know whether your unknown has endospores. One of the ways that you can detect the presence of endospores is through microscopy, and even oftentimes you can see them in unstained preparations. This is an unstained preparations. And you see these refractory areas right here, these clear areas right here, both outside and inside the cell. The ones that are outside the cell have been released when the vegetative cell is broken down and died. Okay? But it's very important in identifying some unknowns to know the location of the endospore within a cell. So sometimes you have to kind of look around and find an intact cell that has an endospore within it. Staining usually facilitates your ability to see endospores. Endospores don't take up the stain nearly as well as the cytoplasm of the vegetative cell did. We had two different bacillus slides out here yesterday, and I did that intentionally because I just wanted to see what they both looked like. And one tended to be longer rods that were chains. I think the majority of the slides were that. But there were some that were a little more short, stubby bacillus bacteria, some of you might have looked at. And if you saw them, most of the cell was purple, but there was a lighter area inside. That was the endospore. Okay? It doesn't take up the stain readily under normal staining conditions. Here is a fluorescent antibody stain preparation showing the development of an endospore on one end of the cell right here. This gives you a better idea of the endospore and how it develops in the cytoplasm of the cell. One of the things that you can see by looking at this micrograph is that the spore is a multi-layered structure. Basically what happens when the endospore forms is the cell segments off a piece of the cell cytoplasm and also the copied DNA of the cell. So prior to endospore formation, the single circular chromosome that makes up the genome of the bacteria does have to duplicate. And those two molecules that are duplicated do have to be pulled apart from one another. One has to be put into kind of a different packet in the cytoplasm of the cell. Do bacteria use spindle fibers like eukaryotic cells do to segregate their chromosomes? No. We think that the mesosome actually plays a role in separating that duplicated circular chromosome from its copy in preparation for endospore formation. And then the cell begins to layer down all these layers of protective material around the cytoplasm and DNA of that segment of the cell, and that forms the endospore. And we'll talk a little bit about what these layers are, and particularly what is it about this process that makes the cell a little bit more resistant for the endospore. Okay, here's another endospore staining technique, and this is the one you're going to use in lab. This is called the Schaefer-Fulton technique, and basically it involves staining cells initially with a green stain called malachite green. You can always tell when we're doing endospore staining because people walk out of the lab with green fingers. In order to get that stain not only into the cytoplasm but into the endospore itself, we have to steam the cells, and this involves making your slide and then putting a little piece of bibulous paper or paper towel on top of this preparation of material to put on there. And then you turn on your Bunsen burner, turn it upside down, and direct the flame down onto that puddle of stain. And as it starts to steam and evaporate, that drives that stain into the endospore itself. You have to keep replacing stain. It takes about 10 minutes to do that steaming procedure. And then what you do after that is you wash the slide with water, and the water wash is sufficient to wash the stain out of the cytoplasm of the cell, but the stain will be retained within the endospore. And then you do a counter stain with safranin, and that again will stain the cytoplasm of the cell, but you're not steaming this time, so that won't stain the endospore. So when you're done, you should have red cells with green endospores either inside or typically outside the cell, because you can imagine what havoc steaming plays on cell integrity. And that's going to cause a lot of the cells to lyse and break down. So you'll see a lot of free released endospores, but if you look around, you'll be able to find endospores within the cell. And then you can do important things like determine their location. Are they found at one end of the cell? That's called terminal. Are they found in the center of the cell? That's called central. And also, you very likely will have to measure those endospores. Because one of the breaks in the Verdi's manual dichotomous key is endospores greater than 0.9 microns in size, endospores less than 0.9 microns in size. Identifying the genus Bacillus to the genus level is pretty much a piece of cake. People who have unknowns that are gram negative, especially short gram negative rocks, have a much more difficult time getting their unknown to the genus level than somebody who has a bacillus. But once you get to the species level of bacillus, it becomes quite challenging because very small differences in endospore location, for example, or size can really put you in different parts of the key in terms of your identification process. So some are more difficult at the genus level, some are more difficult at the species level. So why are endospores resistant? Well, an endospore has a cortex region, which basically is DNA and dehydrated cytoplasm. And then that cortex is surrounded by multiple layers of material that help to just add to the resistance of that particular structure. And there's names for all of those. There's the exosporium and things. I'm not really particularly interested in that. But it is important to understand that this cortex is a very, very dehydrated, almost gel-like cytoplasm, and then DNA. And there's some ribosomes too, obviously. There's enough stuff there to basically make a functional cell. Anybody know what the typical water content of the cytoplasm of a cell is? Small part. How much of your cytoplasm of your cell is water? Yeah, 70, 80%, you know. Cortex, 10 to 15% water content. Very dehydrated. Well, that really tremendously slows down metabolism to have that little water available. So, Zach?
So when it's saturated with water, is that a signal for it to start growing again?
Part of the process of growth involves it taking in water, yes, and then rehydrating. Yep, absolutely. Slower metabolism means you use food resources much more efficiently. You produce less toxins. I mean, metabolism can almost be at the point where it's just almost non-existent. But the cell is still alive. And the endospore material is still alive. Also, one of the reasons that particularly heat is effective in killing cells is that it denatures proteins. Protein denaturation is much more effective in an aqueous environment than a non-aqueous environment. So the presence of water helps to facilitate denaturation of proteins. So dehydrating the cytoplasm really helps to protect those molecules from denaturing. One of the substances that's responsible for helping to bring about this state of dehydration is calcium dipicolinate. And you find a lot of this in the wall material that surrounds the cortex of the endospore. And again, anticipating the question, I've never really seen a great explanation for how the presence of this particular substance contributes to the state of dehydration. But there is a high level of correlation in that particular instance. There are some other molecules that play a role. The cortex is full of a category of molecules called SAPs, small acid-soluble proteins. And these particularly seem to confer protection against radiation. Again, mechanism, unsure. But the more of this molecule there is, the more resistant those endospores are to radiation. The process of endospore formation happens pretty rapidly. Usually it takes about 6 to 8 hours for a vegetative cell to be able to develop an endospore. Germination is even faster. Once those cells are exposed to an appropriate environment, germination of an endospore takes a couple of hours, 1 to 2 hours. This particular slide up here kind of shows you the life cycle of both endospore formation on the right and then germination on the left. It shows you how you can see the mesosomes right here, the infoldings of the membrane, are helping to separate the duplicated chromosomes. So here we're forming a terminal spore. This particular structure is called a forespore, and the duplicated DNA is surrounded by wall material. There are multiple layers of wall material that are deposited around that spore. It becomes more and more dehydrated. But if it gets the proper signals of nutrient levels, water availability, then enzymes are activated. And those enzymes start to form cracks in the cell wall. And then water can move through those cracks and get into the spore cortex and cause the cytoplasm to rehydrate, metabolism to kick back up again. And eventually this will then germinate and form a new cell. Most endospore forming organisms are bacillus. Okay? It's very unusual to find endospores within cocci. Here you can, oops, got a mislabeled here. This isn't the cortex. Cortex is this layer out here. This is called the core of the spore. Sorry about that. So here, over here on this slide, in this particular image right here, you can see the multi-layered nature of that endospore with the core, the cortex, the spore coat, the exosporium. Just worry about the core, okay? Some other things that you may find in the cytoplasm, depending upon the species of bacteria. Are various granules and inclusions. Granules tend to be conglomeration, sorry, can't say that word, groupings of inorganic molecules. Inclusions tend to be groupings or conglomerates of organic molecules. These are kind of storage areas. Inclusions sometimes will have a membrane around them. Granules typically do not. There are various stains that we can use to determine the presence of inclusions and or granules. For example, one of the stains we're going to use is a stain called a PHP stain. Basically, this is a fat stain. PHP is a fatty molecule. PHP is polybeta hydroxybutyric acid. This is a way that bacterial cells can accumulate fats for later use. Just like we can put on fat, but we have specialized adipose tissue to store the fat. They can just store this in the form of lumps, basically, these inclusions in their cytoplasm. We're going to do a PHP stain. And the reagent we will use will be taken up exclusively by the fat granules, the fat inclusions, and they'll stain a dark black. And so you'll actually see black granules within the cytoplasm. Pretty easy to see. Other inclusions that we might see are glycogen inclusions. What would be an easy way to detect the presence of a glycogen inclusion? What's another word for glycogen, do you know? Sometimes it's called animal starch. Okay. It's a polymer of glucose units, like starch is. It's a little bit more branched than starch is. What's the starch test? Iodine? Yeah, you can detect glycogen granules with an iodine test. So just put some iodine on your cells, and you'll see those blue-black granules indicating the presence of glycogen. Sulfur granules are not uncommon in the cytoplasm of some cells. Particularly photosynthetic organisms that carry out a type of photosynthesis called anoxygenic photosynthesis. What does an mean? Without? So these are organisms that photosynthesize but don't produce oxygen. What some of these photosynthetic bacteria do is instead of splitting water molecules during photosynthesis, they split hydrogen sulfide and produce sulfur, which can combine with other sulfurs to form elemental sulfur. And so we can see the accumulation of these sulfur granules in the cytoplasm of these photosynthetic cells. Some cells can then actually use this sulfur as a source of electrons in metabolism, and so they might deplete those granules later on if they need an energy source. Sometimes these granules appear, and then you'll see them shrink depending on what's happening metabolically. Well, I'd hoped to get through everything today, but we didn't quite do it. So we'll wrap up a little bit on Monday with some final cell structures. And then we're going to get into the next chapter, which has to do with bacterial taxonomy, looking at the bacterial groupings, and introduce you to Bergey's manual, which is good because next week in lab, you're going to get your unknown, and we'll talk about the unknown identification process and the process of scheme. Okay? So I'll send you out some things today, just kind of reminding you what in drop on you should be looking at next, and some recommendations for next week. Okay? So exercise number 2 tomorrow. Chapter 15.
So, say they need extra when you test it, will you still see some sulfur? Or
If they had depleted the sulfur granules, they could get to the point where they were completely depleted, and then yes, you would see it.
So then you could have
Mhm.
Are we gonna have?
No, I mean, some of these, we'll do this particular one right here, and we'll be looking at nice healthy cells, and so if they can produce the stuff, then we'll see it. But good question.
Dr. Browder, I'm gonna go get the card.
Oh, okay.